Using X-ray methods, one of the earliest findings was that many folded proteins contain regions that are twisted into helices. A helix is best illustrated by a metal spring, as shown on the right. It is a coil that is stretched out along an axis. By building models of polypeptides, it's been possible to explain why helical foldings are common. If the protein is twisted in just the right way, it's possible for hydrogen bonds to form between all the CO and NH groups along the backbone of the polymer. The hydrogen bonds are shown on the left by means of broken red lines. A hydrogen bond is an attractive force that occurs when a hydrogen that has some positive charge is brought close to an oxygen or nitrogen atom that has some negative charge. The hydrogen bond is only about 1/16 as strong as a typical covalent bond, but the many hydrogen bonds that occur in a helix are collectively strong enough to hold the polypeptide in the coiled form. As you might imagine, there is more than one way to coil a protein to give a helix that is stabilized by hydrogen bonds, but there is only one pattern of folding that allows all the NH and CO groups along the backbone to form hydrogen bonds. This best of helices is called the alpha helix. Other kinds of helices are rare and are given different Greek letter names, such as the beta helix. Various kinds of proteins differ greatly in how many of their amino acids are involved in alpha helical foldings. In most proteins, there are isolated short regions of helix, separated by regions that are not as regularly folded. At the opposite extreme, there are some proteins that are almost all alpha helix. This is true of proteins in hair, for example.